Thanks, Natasha, for the introduction, and thanks the organizer for giving me the opportunity to present our work. Before I give my main talk, I added one page yesterday about protein interaction networks and applications. I worked on the yeast physical protein interaction starting around 2000 when the yeast two-hybrid system was used for generating yeast physical protein interactions. So at that time, the yeast two-hybrid system was initially used to generate all those large-scale protein-protein interactions. So the first question we asked is, how do we estimate the fraction of true protein interactions within the observed interactions, because we have a lot of noise there. So we observed that for true interactions, the correlation between those gene expressions for those interacting protein pairs tend to be higher than those no interacting pairs. Based on that idea, we built a first mixed model to estimate the fraction of true interactions there. This method is very, very simple, and it just uses gene expression to estimate those fractions. So on the other hand, about 2006, I received an email saying that, oh yeah, your method is the best to estimate those fraction of true interactions within that. So, and I was kind of quite confused, and what? It can't be, because it's so simple, it's so easy. And but after about 2 or 3 months, I saw one paper from Tree Deck, and also wrote that Sharon was part of the paper to compare all these different methods to estimate those reliability of those protein-protein interaction network. That includes several different high-profile papers from Nature Biotechnology, PNAS, and so on, and including very fancy methods like supporting vector machines and random forest, and including many different information there. And through the independent validation criteria, they said, okay, so finally they get a weird conclusion, and the method using this gene expression, this mixed model, tended to be the best using independent criteria. And they say, okay, so they say, okay, sometimes simpler is better. Rodent was on part of that paper, and I was surprised to see that one. And that's probably reality is in our research, is if it's use too many information, and sometimes whether it's generalizable or not, that's a big problem. So after we did that work, and the second question we ask is, how do we predict protein functions using protein-protein interaction network? We introduced the idea of micro-random field to predict protein-protein interaction, protein functions based on those protein interactions. And we also integrate many different information, such as domain information, sequence information, gene expression data, and so on, to get a general micro-random field theory to predicting protein functions. And as far as I know, those it's the first so-called message propagation to predict protein functions. And this is also the basic idea about how do you predict the prioritize genes related to complex diseases and so on. After this work, Bill Noble used diffusion kernel over the networks and try to predict the protein functions. And there he showed, oh yeah, this method is better than the micro-random field. And at first I was not convinced just by look at the papers, and I asked the students to reproduce the result. Finally, I say, okay, I have to I was convinced that diffusion kernel is better than the micro-random field. And so since that time, I essentially just like diffusion kernel, and I combine the diffusion kernel together with the micro-random field, and then we get another paper that related that. But it's on the other hand, it's that paper had a difficult time to get published, because it's the accuracy is not really increased compared to the diffusion kernel using different kernel approach. So, but on the other hand, because I like the different kernel and I use that in a variety of different problems, including integrating the gene expression data and the protein-protein interaction data. So there we use the basic idea that if you have differentially expressed genes in the neighborhood and then you integrate that particular gene expression as well as its neighborhood, and then you can predict the differentially expressed genes much better. And so that's it's one that we call it CGI, and that's by Xiaotu Ma in 2007, and that integrated the gene expression and the protein interaction network. We compared that with different kernel with shortest distance as well as direct neighbors, and then we show that the different kernel has the best performance. And the other problem that we did is on the predicting the RNAi the target, and so we know RNAi is you can just knock out using RNAi knockout and try to see the direct target. They are false positive and false negatives. And then we use those idea of different kernel together with the protein-protein interaction network, and we show that there you can just get the true positive from the RNAi experiment. Again, we showed that the different kernel has the best performance in terms of the RNAi experiment. And recently, I see that Shirley Liu also used those basic idea of neighborhood and protein interactions, try to predict the CRISPR target, and that's just published recently last year on the genome biology. So I think it's, as you all know, that you have used different kernel quite a lot and in prioritize genes, and I think it's through all those applications, I think it's different kernel is the way to go, at least for the biological applications. I had been trying to promote those different kernels in many different situations, and the common question from mathematicians are, can you mathematically prove that different kernel is the best? And I feel a little bit hard to do that. I don't know how do I answer that kind of question to say the different kernel is the best for the network-related stuff. I really don't know. But I think it's why the different kernel is the best way to solve those kind of biological problem. My basic intuition is different kernel probably really capture how the information is transmitted through the network. I think that's probably the way, and I don't think it's mathematically it's possible to do that. Another the project that gets quite a lot of attention, including Teresa, that use those random walk and the network stuff and try to infer the pathways from the mutations to the target and so on. And so that's about all this related on the random walk on those networks. I think that's part of those our common effort. And we have know all this noise and this shortcomings, criticism of this network, but through all this analysis, I think at least I'm convinced that network is useful for basic biological studies as well as for disease gene prioritization and pathway inference. Other problem that is not touched on in this symposium is we also developed some maximum likelihood approach for predicting domain-domain interactions from protein interactions, and that actually is one of my most widely cited papers among all my publications. So I also developed some method for identify network motifs in those random networks. So that's about those networks, and in the East, not in the human. Okay, so now let me get to my main topic on the local similarity analysis of time series data and the applications to metagenomics. Again, this is different from the main theme of this talk, and I think at the last minute, it's probably you want to hear something, not just the disease. So how this project started? Actually, it's about 2005 or something. I heard a talk about those metagenomics, and then they talk about all those in the waste treatment, and there are all these different organisms and so on. And now it's talking about system biology. We not only just consider ourselves, we also consider all these metagenomes, different microbials, and so on. And then I get quite interested in that, and so I try to figure out who in University of California work on this kind of problem. Fortunately, we have one expert in this project, this Professor Fuhrman, who have been working on the Observatory for the marine system for quite some time, studying the 1998. And they collect water samples from different locations of the world, as well as time series data. Every month they collect water samples from the ocean, and then they try to understand how the environmental factors affect the composition of those communities, and also how those microbials just associate with each other, and so on. And at that time, there was no next generation sequencing data available, but they, using some other techniques, try to identify what kind of organisms are there. So then they say, okay, so they have, at that time, they have about 10 years data. They try to say, Fengzhu, what can you do with this kind of data? And how do I study the relationship between these different organisms? So that's how the project started, and you can see they get those different locations. In particular, it's the Catalina Island, and then they also get outside of this Catalina Island to different part of the world, and then they also get different depths. And so it's a real great data set of spatial temporal data, and then we try to understand what is going on. So now we have 18 years of spot data, and after that we also get involved with the dimension of diversity project, that they try to study how the genetics and also the environmental factors and ecology that just affect diversity of the ocean. So that's we had a joint project together on those kind of data analysis. With the next generation sequencing data, now we have more techniques to identify what organisms there, and we can get a more refined scale about the organisms in those communities. And now we can study the similar kind of question, get to the more finer level about those interactions between those organisms.
Can you say precisely what is this time series data? This time series data is...
It's monthly data. It's every month.
What kind of data is this?
Okay, so they collect the biological as well as the environmental factors, and then they get the water, and from the water you can if we use next generation sequencing data, you can get the 16S gene sequence, and through that sequence you can do clustering, and then you get something called the operational taxonomy unit, and that's the code, the representation of the organism. That's the kind of data that we have. So essentially we have the abundance level of the so-called operational taxonomy units. And for those who are not working on ecology, you just think, okay, this is the part of the organism, okay? And then we just try to study the relationship between those organisms, or in the...
Environmental factor, you mean temperature and pressure and...
The temperature, yeah, temperature, sunlight, and so on. And also some biochemical. They measure a lot of how the water, the function properties of the water system.
So it's at a fixed station?
Huh?
It's a fixed station, or it's now floats?
I mean, that cannot be guaranteed. that cannot be. But they get the same location, but water just flows and then see certainly the metagenome, metatranscriptional data too. Okay, so these are the scientific questions that we try to ask. It's now let's see when it's 16S data, and then we have the 16S essentially is a it's a conserved gene part that you can use those gene to see what organisms are there, but it's definitely not exactly, but we try to bin them. And then using the sequence data, so that's called the tag sequence. And then from there, we try to classify those genes, the sequences, and then we get the different cluster. And the cluster itself, each class we call it operational taxonomy unit. Okay, so yeah, this is just as a jargon that ecologists used for represent that. And the definition of operational taxonomy units actually is not precise, and actually there are a lot of debate. How do you define operational taxonomy units? There are still algorithms developed. How do you define that, and so on? With very large number of sequences, how do you define that? It's not trivial because of the sequencing errors and so on. There are the early studies show that, okay, so they get thousands of rare organisms, OTUs in the ocean, and then there's a so-called scientific hypothesis. There are rare space sphere and so on, biosphere and so on. But later people say, oh yeah, it's not. And even if you put some 40 species into the water, and then you just sequence that, you can get several hundred of species there. But it's so again, it's define OTU itself is not a trivial thing. But here, let's assume that the OTU is already defined. Okay, I'm not dealing with how do you define OTU, and suppose the OTU is already defined. Okay. So now the OTU is defined, and then we get the OTU abundance level in each community. And so at first, I mean, I say, okay, this is the easy problem, and we do this gene expression analysis all the time. Let's do correlation analysis, get a network analysis, and so on. And that's what I thought is just quite so simple. And so that's what I did. And then it turned out some of them give you meaningful result, but if you look at the hot clustering, and then you will see some part of them you can align quite well, but some of them essentially is just like random, and you don't see there is a pattern there. And then we try to see what is going on there. Actually, it's if you just look at it carefully, and you see during the winter time, a lot of these organisms are not abundant at all, and there are no correlation between those organisms in the winter time. And to me, it's a no biologist. I felt, oh yeah, this is what is going on. And at first, actually, I made some mistake. I say, okay, summertime, and actually, the time you really see the correlation is sometimes from March to sometime October. And that's the time you see the correlation. And from the March time, then you don't, like January or December, you don't see a lot of correlation there. And what is going on? And I say, okay, so at first, according to my naive knowledge, I thought the spring should be from January to let's say to March, and it should be January, the springtime, and that time. But actually, it's not, right? And finally realized, oh yeah, my naive knowledge is really wrong. And so, but it's really meaningful that you can see the correlation actually is from spring around the March time to October to November sometime. And so the time during the winter time And even if it's in the ocean, you don't see anything there. It's just like our system, and many of them just dominate there, and you don't see the correlation. So that gives us something that when we study the correlation, it's not the best way to just use this PCC to just to say, oh yeah, this is high, and then we get the correlation, and if it's not high, we don't get correlation. Let's think about those. If you are given a time series data, if you just look at the correlation, and if most points are not correlated, then PCC will not work. So now in that sense, it's how do you really define the correlation between those time-dependent correlations? So that's what we want to study. So now that's getting to two definitions that we try to define. One is so-called local association. What do we mean by local? Is that if we have a time series data, and some part of that, like in this simulated example, we simulated around 20 data points, and then from the first one to the seventh, we just random simulated, and from this 17 to the 20, and we also random simulated, but there are some correlations between them only in this time interval. So that's try to simulate the point that we are seeing in our real data. So the other thing that from biological point of view is there are some delayed associations, and so we know this factor is going up, the other factor is going down, and so on. And so there are some delays instead of just look at those point-to-point correlations. So in this example, and we say that if we we have this one, and in this one we do a time delay, and if you move according to that time delay, and then you see a high correlation between them. But if you just purely calculate this correlation from here, then it's just you are not able to find those correlations. So the scientific problem we try to do here is giving a time series data, we try to find such correlations that you have, maybe it's local, maybe it's time delayed, but we don't know where those the intervals are and where those delays are. But it's for practical purposes, we want the delay to be just within a short interval. If it's too long delay, and then we just say, okay, we are not interested. Okay, so now we essentially just get into the data, and now essentially the data is if we have two, and then we have those x1, x2, xn, and the second data is y1, y2, yn. The first thing we do, that's a statistical trick that we just normalize that using those rank things, and then we get the normalized. And although this is the very simple trick, but actually in practice you will see that this very simple trick can be quite useful in practice. Because if you purely use those data, a lot of this data the normality is not satisfied. And then you probably cannot detect it. But with this simple one, sometimes you can get better result. Okay, so now with this data, you mathematically we try to find those subinterval of length L such that you have this one, and they start from I and start from J here, and then with those I minus J, the starting point less or equal to D. D is whatever you defined according to the biological knowledge, what you try to find, what kind of delay you are interested, and then you try to maximize this score. That's essentially what we try to get here. And so once you see this one, you say, oh yeah, that's essentially it's similar to like the local sequence alignment, and that's exactly what we the first thing that we do. Actually, this is not completely new from our group. Actually, Mark Gerstin's group used this basic idea to do the correlations for gene expression data, and he did this. And for this audience, I think everybody knows this. I'm not going to talk about it. Okay, so then after we did this work together with Jed Fuhrman, and he liked it, and he talked about it yesterday. And then he talked about it, and then he showed that many of those people in the community try to use this program. And fortunately, and then one of his collaborators raised the following question to us. Now, suppose in each data point, you not only just get one sample, you get multiple samples. And what can you gain by multiple samples? Actually, he collected those kind of data, and then he tried to write a grant proposal on this type of data. And that's essentially give us some idea about how do we do this. And this is a good question. And then we just get this, and then we see, okay, so you summarize data, and then we do the Bootstrap confidence interval for the local similarity score, and then we also get the p-values statistical significance for this one. So, honestly speaking, at the first I just thought, okay, we just permute the data, and then we see that should be fine. But it's then later as the next generation sequencing data getting more and more, and then it takes several weeks or a month to really get these p-values. And Jed said, Fengzhu, you have to solve this problem. I cannot use your program. I will not use your program if you don't solve this problem. And so then I have to solve this problem. I communicated with the mathematicians. And then it's how do we get the theoretical p-value for this local similarity score? And you might say, okay, so the local alignment score, we have some Collins great work for the extreme value distribution. Why can we not use that? And the reason for why we cannot use the extreme value distribution is because in the local alignment, there is the condition, the expected score should be less than 0. And here we have the expected score equal to 0. Okay, so like people like being probably will say, oh yeah, I know how to solve this problem, but for me, I don't know how to solve this problem. And I ask the mathematicians, how do I solve this problem? And honestly speaking, it's just take a lot of time to understand what I'm talking about, what he talked about. Finally, he gave me this paper that I really like a lot, and Feller is one of these great probabilists there, and he got those, studied those excursion of those random walk with expectation equal to 0. And that's kind of that we break the idea, and then we use this idea to get the theoretical p-value. And so you can see it's not easy, and I really appreciate those great mathematicians to solve this problem. And honestly speaking, I cannot solve it by myself, even at this moment, if I try to prove myself. But he's used very mathematical theory to get those density function, and the density function is a derivative of this complex function here. And after that, I can just get this infinite series to get the p-value. So anyway, so that's.
Is this your result? This one?
This is not my theorem. It's a Feller's theorem. Yeah, it's Feller's theorem. But what I did is just from here to here. That's what I did. I'm trying to. Honestly speaking, I'm a middleman, and just to get this biological problem, and then connect to the mathematical problem, and then I did some simulation, try to see whether and what kind of we try to see is it okay to use this one, and if I have time series of 20 of I don't have enough time. I just say this is the I did the simulations and try to see what these are the simulations for the histogram of that local similarity score, and this is based on the Feller's theorem. Okay, and so you can see it's not perfect, but at least for those tail probabilities, that's what I'm interested, and that seems to be okay. And here is okay. I also just see what is really what really okay means, and so I just say okay. So if the number of time point is 20 until 100, and I see it's what is the p-value through those theory here, and also what is the simulated p-value here, and then you can see here it's here. I mean, what we are really interested is at least it's probably is less than 0.0001 or something. It's a very small p-value, and you can see here it's actually quite close. And the most important thing is that this theoretical value is conservative. It's in the sense that theoretical p-value is a little bigger than the simulated p-value. So I'm good there. And in this sense, essentially, I can do a hybrid approach. And if I use this theory, I can get a p-value. I cut the threshold there. It's a little bit higher than that. Most of that result will be just cut out. And then if I want to get a very accurate p-value, I can just do simulations on those left pairs. And that's solve the problem. Okay, good. So I'm done. And then let me just see we have the software, and then there are some competitions, and that has nothing to do with me, and that's actually is by Rob Knight organize those things, and they collect a lot of those different methods, and actually they generate the interaction data, and then they let different labs to see what you have. So that's the final result that they get, and I did not generate this figure. That's from Rob Knight's group, and so this just come out from the ISME journal, and so essentially that's a suggestion, and you can see here it's the LSA, and this is the LSA, and this is the LSA. And so that's our local similarity analysis. No other method shows up except that they say, okay, so you integrate all those different methods. Sometimes it's getting a little bit better result, but actually the final conclusion is LSA actually is doing quite well. And we know the maximum information content is, a lot of criticism already for that one, but actually it's not that bad in some situations. Okay, so I think I just done. Thank you.
Let's take a couple of questions.
Yeah. So in the local association that you're looking at, so you say it's like a sequence, like
it's like a
no gaps, right?
No gaps here. Yeah.
So how many years did you collect the data?
Okay, so the data was collected over now is starting from 1998. So it's the purpose, the whole purpose, they just definitely is not really designed for this study. They just try to study the data, those kind of stuff.
I'm wondering that, did you see the data like same month, different years?
It changed somewhat. Yeah, it's because of the temperature. It's not like.
Less than the all year round.
Yeah. It's not always. March this year is different from March last year. Yeah.
How fast is this Fehler's method for computing? How fast?
Because it's theory, so it's a theory. It's nothing. Yeah. So that's what we see. Okay, probably that's I considered my major contribution there.
Final question? Yeah. Have you assembled or just you use the reads itself?
Huh?
Have you assembled the reads or you just use
Read the result. Can you repeat your question, please?
So when you have sequence, do you have reads?
Yeah, we have read. Yeah. Okay, so it's really depend. It's for this particular problem, read assembly is not the key issue because here they usually have very short readings. And so, but it's clustering of the sequences, that's the challenging part. Thanks.
Let's thank the speaker again.